This is the second half of Chapter 9 where we're talking about oncogenes. And in this part, we're going to focus on specific examples of oncogenes. And as you probably got the idea from the first half, there are actually lots of different oncogenic signaling pathways in cells, which could be hugely overwhelming. So for example, in this one diagram, you can see a bunch of different cell signaling pathways that would all potentially lead to cancer if they became oncogenic. Um, and I know you're probably familiar with this basic structure of a signaling pathway, but you've got, ligand signaling molecules coming from outside the cell, binding to specific receptors on the cell surface. Those receptors change their shape upon binding to the ligands, and those shape changes then activate molecules in the cell, which activate other molecules in the cell. So I always like to use the analogy of kind of a relay race here, where one molecule is passing the baton off to the next molecule or runner um, throughout the race. And then, as you can see in this example, most of these signal transduction pathways are all headed towards the nucleus, where we're activating transcription factors, which are then going to turn on transcription of specific genes, causing the production of new proteins, and then specific changes in biological activity. So we could spend a lot of time talking about, all these different signaling pathways that have been linked to different cancers. But I want to instead just focus on one particular pathway, the RAS pathway. And my feeling is if you can have a good understanding of that pathway, you could really look at any other signaling pathway and understand how it works and how overexpression or hyperactivity of certain proteins could lead to cancer. So we'll look at that RAS pathway, and then I also just want to talk a little bit at the end of the chapter here about how oncogenes could really appear at any of these levels throughout these cell signaling pathways, any of these stages. Okay, so let's look at the RAS, sometimes called the RAS-MAPK pathway. And so, as we just discussed, right, this pathway starts with a ligand, in this case, growth factor, binding to a receptor. The receptor for the RAS pathway is what's called a receptor tyrosine kinase. And this means that the receptor itself actually has kinase activity. So remember, kinases are enzymes that can phosphorylate other proteins. And these receptor tyrosine kinases usually exist in two pieces, and binding of the ligand, the growth factor in this case, will bring the two halves together and activate that receptor. And so once it becomes activated, it will start basically gathering phosphates on the portion of the receptor that's inside the cell. Right? This is a transmembrane protein. We've got the portion that interacts with ligand outside on the cell surface, and then this other biologically significant portion in the cytoplasm inside the cell. So once activated, it gathers those phosphate groups, primarily from ATP, and it will then use those to activate these adapter proteins, which will then activate the namesake RAS protein. And you'll notice RAS is here, it's a peripheral membrane protein associated with the inner surface of the plasma membrane, and it's what's considered a G protein. And I want you to think of these G proteins as basically on-off switches. So when RAS is inactive, as it is up here in the top image, bound to GDP, guanosine diphosphate, the switch is off. But if it becomes activated, bound to GTP, guanosine triphosphate, now we're flipping that switch on, and it will in turn activate other molecules in the cell, primarily this kinase called RAF. And so just to focus in here on RAS for 1 second. So again, it's really a molecular on-off switch. So active bound to GTP, inactive bound to GDP, And we've got these two classes of proteins. These really apply to any G protein that are turning that switch on and off. So there's the GEFs, which stand for guanine nucleotide exchange factors. So we're basically exchanging GDP for GTP to cause that activation. And then you have GAPs on the other side, which are GTPase activating proteins. GTPases are enzymes that chew up GTP. So if we activate a GTPase, we get rid of the GTP and we're back to GDP and we flip the switch off. So flip it on, flip it off. If we have these gaps or GEFs present, we can do that very quickly. Right, so you get the idea. These adapter proteins here that are being phosphorylated by that receptor fall into this GEF category. And then they're going to activate RAS, which will then in turn activate RAF, which is a kinase, so it's going to phosphorylate other molecules. And at this point, we're going to move into what's called a kinase cascade, or sometimes a phosphorylation cascade, which is where you have one kinase activating another kinase, activating another kinase. So in this case, we've got the RAF kinase phosphorylates this inactive MEK kinase. Once it becomes phosphorylated, it's now active, and it will phosphorylate the inactive MAP kinase. Once it's active, it will phosphorylate transcription factors. We've got sort of two branches here, ETS and June. But really the important point here is we've moved this signaling pathway into the nucleus where we're starting to activate transcription factors. We see MYC get activated. We've already talked about MYC. E2F is another transcription factor we've mentioned in previous chapters getting activated. But the end result here is going to be to start transcribing cyclin CDK genes. And remember, those are the proteins that are important for getting the cell past those various checkpoints throughout the cell cycle. So that's going to increase cell division, movement through the cell cycle. Okay, so we've got a lot of steps. And I think it's important to point out that there's really two reasons that these signaling pathways typically have so many steps. One is large number of steps kind of amplifies the signal. So it'll make the signal bigger and louder as you move step to step through this relay race. And the other reason is it gives the cell a lot of various control points. So more steps means the cell can really kind of fine-tune the intensity of the signal and hence the response. And we'll come back to this at the end of this second half of this lecture. But I did just want to point out now that really any of these proteins that are part of this pathway could potentially become oncogenic, right? A lot of these would be considered proto-oncogenes. So even though we're focused on RAS here, and RAS is certainly a proto-oncogene that often becomes oncogenic, RAF could be a proto-oncogene, MAPK could be a proto-oncogene, some of these transcription factors could be proto-oncogenes for that matter, and we'll come back and talk about that idea again just briefly. Excuse me. So that RAF kinase, as well as the MEK and the MAPK, are all examples of what are called serine-threonine kinases, just meaning they like to add phosphate groups to serine and threonine amino acids in other proteins. But there is another big class of kinase that are called the NTKs, non-receptor tyrosine kinases. So you may remember the receptor that we had in this RAS pathway was a receptor tyrosine kinase, likes to phosphorylate tyrosine amino acids. But there are some of these tyrosine kinases that aren't receptors. And there's some very well-known examples here. They're parts of other pathways. So there's the SRC kinase, the JAK kinase, ABL kinase. You might remember we saw that in the BCR-ABL translocation gene fusion. So just tuck that away and keep in mind, there are some other very, very well-known and biologically important kinases. Also want you to think about the fact that even though we're focused on, let me go back here, this very well-defined signaling pathway and the fact that, MEK is phosphorylating MAPK, which is phosphorylating these two specific transcription factors. Even though we have that narrow focus, these kinases may be doing a lot more than that, and evidence suggests they are. So, for example, in this particular experiment, you're looking at a protein gel, and they're using a technique. So each band here would be an individual protein. You can separate proteins on a gel just like you would DNA molecules. They're using a technique here that allows them to visualize phosphorylated proteins. So if you see the protein on the gel, that means it's phosphorylated. And what you're looking at here is you've got a control where there's no phosphorylation, and then in the sample on the right here, you've got a single Src kinase. So this would, let me go back, right? This would be one of these non-receptor tyrosine kinases. So one Src kinase that's being overexpressed in that cell, and you can see there's a ton of proteins that are getting phosphorylated, right? So probably the same thing is happening back here, right? MEK is probably phosphorylating a lot more than MAPK. So there can be pretty diverse biological effects if one of these proteins becomes hyperactive. Now to finish our deep dive here into the RAS pathway, I just wanted to focus in on what's going on here in the nucleus with the transcription factors, and point out that we have these kind of waves of transcription factor activation. A lot of times you'll see what are called early genes and late genes. This is really common to have at least kind of these two rounds of transcription factors being activated. So let's, as I already said, right, in this case we're aiming towards production of cyclin and CDK proteins through the activity of these transcription factors. Sometimes you'll see these two rounds of transcription factors being called immediate early genes or delayed early genes, or sometimes they'll be called early and late genes, but it's the same idea. And so what's really happening here is you have this signaling pathway, right, that we've already talked about, leading to the activation of a transcription factor in the nucleus. That transcription factor now goes and turns on transcription of a set of genes, the immediate early genes. They're transcribed, the mRNAs leave the nucleus and get translated out in the cytoplasm. And it turns out all of these immediate early genes are simply producing a second round of transcription factors. So those transcription factor proteins go back into the nucleus now and activate the second round, the delayed early genes. And this might seem a little crazy, like, okay, if the cell just wants to activate these genes, why doesn't it just do that right off the bat, right? Why have this, this 1-2 step to get there? And really the reason is the cell is intentionally trying to create some space, some time to get something else done. So temporal separation would be the fancy way of saying that. So, if you imagine a cell that's getting ready to go through mitosis, for example, it may need to give the cell some time to copy all the DNA, right? Make sure we've got the full copy before going into mitosis. So, these delayed rounds of transcription is basically buying the cell a little bit of time, so to speak. So it is intentional and does serve a purpose. All right, the last thing I wanted to point out specifically with RAS here is that RAS is actually sitting at the top of at least 3 different cell signaling pathways. which is probably the reason it was the first oncogene identified, because any of these three pathways could actually be involved with cancer. So we focused on this central pathway here, right, with our RAF and MEK, etc., leading to production of cyclin and CDK. But there's another branch over here that RAS can activate that's going to lead to rearrangements in the actin microfilaments that are going to help drive cell movement. And of course, cell movement and migration is a common hallmark of cancer cells. So if we overexpress RAS or becomes hyperactive, we get too much cell movement. And then in the pathway over here on the left, this can also help increase cell division, cell growth, but can inhibit apoptosis. So really just with a single oncogene, we're inhibiting apoptosis, stimulating growth, stimulating the ability to move past these cell cycle checkpoints, stimulating cell movement, right? All of those are not good. So it gets complicated, right? All right, so as I mentioned, to finish this out, I just wanted to talk about the fact that in any of these cell signaling pathways that could be oncogenic, we could really see oncogenes throughout the pathway. So we were focused on RAS primarily in that example, but even the very signaling molecule that triggers one of these pathways, the growth factor, for example, could be oncogenic. So just to give you one example, dealing with platelet-derived blood factor, if you have a wound, blood platelets accumulate, start producing this platelet-derived growth factor that are going to bind to cells in the vicinity and trigger these pathways and cause them to start dividing and basically heal the wound. You can see this in a petri dish. You've got a cell with a plate with a bunch of fibroblast cells growing on it, and you create an artificial wound just by scraping through the middle of the cells with a very fine pipette tip. So you've got your wound here, and you can see the difference if we apply some platelet-derived growth factor. We quickly kick on cell division to fill in that wound, versus if we don't, right, the wound remains, started to creep in a little bit here, but very significant difference there. And that's in just 48 hours, right? So this is a pretty quick effect. So you could imagine if you've got cells producing growth factor that are stimulating its own proliferation, this could be an issue, could lead to cancer. We already talked about the fact in previous chapters some viruses are carrying oncogenes that may encode growth factors. That could lead to increased cell division. Translocation, in some examples, is known to cause overproduction of that particular platelet-derived growth factor. So there's lots of ways that could become a problem. So the growth factor could be an issue. The receptor itself could be a problem. This is very common. So in our normal example, right, we have our receptor that is only activated by binding of the signal of a ligand. But you could have a situation where the gene for the receptor protein has been mutated such that it is constitutively active. So even without any signal, and in fact in this particular mutant, right, the protein is even lacking the part that would even bind to the signal, but what's left of that receptor is always on, so it's always sending the downstream signal to divide. Or going back to our idea of gene amplification, right, if we just produce too many copies of the receptor, now we're going to get too much signaling and overproduction of the protein in the cell. So you're probably familiar with this idea, but there's certain breast cancers that are tied in with amplification of the estrogen receptor. So if you have too much estrogen receptor, then the estrogen hormone is signaling those breast cells and some also in some ovarian cancers as well, those cells to divide too much. So growth factor, the signal could be a problem if it's overproduced or overperceived. The receptor itself could be oncogenic. And also just wanted to point out the way that cells would normally limit that receptor ligand interaction. So in this particular example, this is the epidermal growth factor receptor, which is being stained for. So this is the EGF receptor you can see in white here. And of course, it's located on the cell surface, on the plasma membrane. And what they're doing here is introducing a little pulse of epidermal growth factor. So as soon as it binds to the receptor, what you see happen here is the cell basically internalizes all of the receptor. So just sucks it in from the cell surface, endocytoses it into vesicles inside the cell. So it is no longer there to just keep receiving signal. And it's doing that because it doesn't want to receive too much signal. It wants to limit it, and that would be sort of the normal mechanism. So imagine if this particular mechanism was defective, right? The receptor just stays there, then maybe the cell does receive too much signal. And the last thing I want to point out is something that might be kind of counterintuitive, is that you could even have oncogenes in the middle of a tumor suppressor pathway. So in this example, we've seen this image before. This is the apoptosis pathway, right? Either external or internal, depending on whether it's being activated by a signaling molecule or DNA damage. But if we think of the internal pathway, which we've talked about, normally this would be a tumor suppressor pathway because DNA damage would trigger p53, which would trigger the death-promoting proteins, which would poke holes in the mitochondria, allow cytochrome C to leak out, which activates this Cas-based cascade of digestive enzymes, which then leads to apoptosis, and we kill the cell, right? We get rid of the cell that had the DNA damage. That would be a good thing, would help limit cancer. So what does it have to do with oncogenes? Well, if we remember the BCL2 protein here, excuse me, BCL2 is a protein which sits on the surface of the mitochondria and basically fights against those death-promoting proteins. So imagine your cell is overproducing BCL2, right? Overexpressing it. Now we're basically blocking this, right? BCL2 is winning this fight, and we're not going to proceed to apoptosis. So even in the midst of this tumor suppressor pathway, you could see an oncogene. And I'll show you the next chapter when we talk about tumor suppressor genes. You could have tumor suppressor genes in the middle of oncogenic pathways in some instances as well. So thank you for hanging with me, and next chapter we will talk about tumor suppressor genes.